Hello everyone. This video is going to help you review for the DNA test. In particular, it's going to cover DNA mutations. So what is a mutation? It is any change to the DNA code. It can be harmful, it can be beneficial, or it can be neutral. It can change just one protein or the whole organism. Harmful mutations can lead to things like diseases or death. Beneficial mutations are what drive evolution. So we have two main categories of mutations: gene mutations and chromosomal mutations. Our gene mutations are substitutions, insertions, and deletions. The last two are also called frame-shift mutations, and we'll talk about that. Chromosomal mutations include duplications, deletions, inversions, translocations. A lot more complicated. So basically what you have to keep straight is that gene mutations are affecting the gene. They're affecting letters or bases on the gene, where chromosomal mutations are affecting entire genes on the chromosomes. So first we're going to talk about the gene mutations, the first one being substitution. This is where one base is exchanged for another. A letter gets switched around. AUG becomes ACG. So that can definitely change up our mRNA code. It's also called a point mutation because it's only happening at one point. So it's changing one codon. This can result in a new amino acid, but because we have many different codons per amino acid, you could be safe. You could end up having a redundancy situation. Or this can result in a new protein. If you change even one amino acid, and it keeps the protein from folding properly, you can end up with an entirely new protein, and it won't work anymore, or it does a completely different function. So insertion is the next type. This is where an extra base is added. If you notice in the picture here, they take an extra T base and add it in between the T and the G up here. So what is going to end up happening is that every single codon after this base is inserted will be different, because now we're shifting the reading frame. We're changing the three-letter codons that we're looking at. Most of the time this ends up resulting in an entirely new protein or no protein at all, since so many codons are being changed. A deletion is kind of the same thing, except instead of adding a base, we're deleting one. We're taking one away. Every single codon after that will be different because we have to shift our reading frame. So these frame shift mutations, this is another way to look at them. If these are our frames, codon 1, codon 2, and codon 3, they're 3 letters long. Three letters, codon. Now if we add one T in here, it's shifted everything over. Codon 2 is no longer CCC, it's TCC. So instead of proline, we have serine. It's a completely new amino acid, and every single amino acid afterward is going to be different because it's shifted over one letter. You can also do the same thing with deletions. If we deleted a C, now our second codon would be CCA, and that would be an entirely different protein. And just remember that these gene mutations don't have to just be one letter. We can add 2 letters or 3 letters, and those will still shift our entire reading frame. It's true that if we add 3, every single amino acid afterward will be the same, but now we have this extra amino acid that we weren't counting on. That can end up changing your whole protein too. So once we get into the chromosomes, remember we're thinking about genes being added and deleted and moved around, no longer just the letters. We're talking about entire sequences of DNA. So the first one is a chromosome deletion. This is where a large portion of the DNA is removed. It could be 1 gene, it could be 3, it could be 5. It all depends. So this makes your chromosome smaller, and it can even kill an organism if something like this happens. If you're losing important genes that you need to live, you can't live without them, so you won't even really exist anymore if this happens when you're in your mother's womb. If this happens as an adult, it can end up leading to disease and eventual death. So for chromosome duplications, this is where genes are being copied too many times. So in this case, we have a pink and a green, and there's only supposed to be one pink and green, but it accidentally gets copied again. We duplicated our genetic material. It makes the chromosome longer. Usually they're not lethal, but sometimes these can cause diseases. Now, it doesn't only happen once, so this duplication can happen one time, or it can happen over and over and over again. Usually when you have more repeats, you'll end up having more problems. Our next one is a chromosome inversion. When you're inverting, it's like you're turning upside down. So this is where parts of the chromosomes will be turned upside down and switch places. If you look at this chromosome here, all of the genes are in a nice order that makes sense. A, B, C, D, E, F. But if we were to invert the chromosome and they were to all switch places, you may end up with A, E, D, C, B, F. Usually there's no problems with this because you still have all of the same genes, they're just in a different order. They're still on the same chromosome, so hopefully you won't have any problems. The problems are if this inversion doesn't go well, and you end up cutting one of your genes in half, and half of it goes to one side, and the other half stays on the original side. The last one that we're going to talk about is translocation. We're moving locations from one chromosome to a different chromosome entirely. So you have two chromosomes and they're going to decide to swap genetic information. In the case of this picture, one little piece of this blue chromosome is switching places with this tiny piece on the pink chromosome. There's usually no problems, once again, because you still have all of the same genes. You're not missing anything, but with anything genetic, you never know when a small change could end up causing a disease. So our overall chromosome mutations, we can delete. We deleted the letter B in this one. We can duplicate. We copied the letter B. We can invert. We can switch the order of all the bases. And we can translocate, where two chromosomes end up swapping genetic material. We switch our genes. So like I said, there's three possible consequences of mutations. They can be bad for you, they can be good for you, or they can be neutral, where they don't help you do anything better, but they don't hurt you either. So harmful mutations are usually what we think of first. They can end up causing diseases or death because our proteins aren't being made properly. Albinism is an example. Here's an albino peacock. They lose the ability to produce melanin. Another example is like the disease that Stephen Hawking has, ALS. He's not producing enough of a specific protein that helps him control his body. Another example is sickle cell anemia, where our normal red blood cells are changed to look like this sickle shape. Sickle cell anemia ends up causing a lot of pain and problems with your body. Neutral mutations are ones that don't help you, but they don't hurt you either. So for example, if you have one blue eye, one brown eye, it's not really going to help you see any better. It won't help you see worse, but you have a mutation. Beneficial mutations are one that give you an advantage over other organisms. These are the ones that drive evolution. If I'm taller or faster, I'm going to be able to survive better compared to all the other people in the world. Now, I did say that sickle cell anemia was a harmful mutation, but it's also a good one, because if you have red blood cells that have this funny sickle shape, and you live in places where there's high incidences of malaria, you'll have protection from malaria. The malarial parasites can't get into your blood cells when they're this shape, so you're protected from getting a horrible disease. So what I want you to get from this slide is just this particular term, a mutagen. A mutagen is anything that can cause a mutation, whether it's too much UV rays from the sun, or it's X-rays, or it's the chemicals in cigarettes. Mutagens are anything that cause mutations. So what were the big ideas of this whole PowerPoint? A mutation is any change to the original sequence of our DNA. There's two major categories of mutations: gene mutations and chromosomal mutations. Genes deal with one or a few bases. We're looking at a gene. Chromosomal mutations, we're looking at the whole chromosome, so we're going to have one or more genes affected. There's more DNA that's being affected. In gene mutations, you can have substitution, where you switch the letters, insertion, where you add an extra base, or deletion, where you delete a base. In chromosomal mutations, we can have deletion, where we remove genes, duplication, where we add extra copies of a gene. We can invert genes, where they switch places, but they're on the same chromosome still. Or we can translocate, where the genes will switch places between two different chromosomes. Mutations can be harmful, beneficial, or neutral, and anything that causes a mutation is called a mutagen. I hope this helped, and study hard for the test. See you guys later.